السلام عليكم أعزائي الطلبة. Our lecture today in parasitology about liver flukes helm. Liver flukes. Liver flukes are parasites that localized in the liver and biliary passage of various mammalians. Human liver flukes of two familials, the obese Thorxidae and Fasciolidae. The obese Thorxidae has three species: Clonorchis sinensis, Opisthochus viverni, and Opisthochus felinus. In the Fasciolidae, there are two species, Fasciola gigantica and Fasciola hepatica. Fasciola hepatica, which known also sheep liver fluke, and Fasciola gigantica mainly infect of gutters. Causes fasciolitis in human. The parasite vary in adult and egg size and species of the snail. Host of the family Liminidia are usually the intermediate host. Fasciola hepatica was the largest and most common liver fluke found in the human, but its primary host is the sheep. It's worldwide in distribution, being found mainly in sheep rearing area. It causes the economically important disease liver rot in sheep. Humans usually become infected by eating aquatic plant grown in water contaminated with feces from animal harboring fasciola. The adult worm lives in the biliary tract of the definitive host for many years, about 5 years in sheep and 10 years in human. Its large leaf shape, fleshy flukes, 30 mm long and 15 mm broad, gray or brown in color. It has conical projection anteriorly and it's surrounded posteriorly. The eggs are large, ovoid, operculated, bile stained. They are laid in the biliary passage and shed in feces. This figure shows the adult worm, and it has oral sucker and ventral sucker. Also, it has uterus, ovary, and testes. The other figure shows the egg of this element, and it's ovoid, operculated, and bile stained. Life cycle. The adult worm lives in the bile duct of the final host, and immature eggs discharged in the biliary ducts are excreted in the feces of the host. The embryo matures in the water in about 10 days, and the miracidium escape in the and invade intermediate host snail. In the snail, the parasite undergoes several developmental stages: sporocyst, lydia, and cercaria. The cercaria are released from the snail and insist as metacercaria on aquatic vegetation or other surface. Sheep, cattle, or human eating water vegetation containing the metacercaria become infected. After ingestion, the metacercaria exist in the duodenum and migrate through intestinal wall, the peritoneal cavity, and the liver parenchyma into the biliary ducts, where they develop into adult. In human, maturation from metacercaria into adult flukes take approximately 3 to 4 months. This figure shows the life cycle of the Fasciola hepatica. The life cycle began when an embryonated egg passed in the feces of the definitive host, and then in the fresh water maturation of the embryonated egg and released miracidium that penetrate the intermediate host, the snail. Inside the snail, the miracidia undergo several developmental stages, which began with sporocyte and rhidia and finally form cercaria. Cercaria released from the snail and swim in the water and then insist on a water plant. to form metacercaria. Then when metacercaria taken by definitive hosts such as human, sheep, cattle, when eaten water grasses contaminated with this metacercaria, inside these definitive hosts, metacercaria exist in duodenum and then penetrate the intestinal wall and peritoneal cavity and reach to the liver and biliary tract, and their maturation to form adult worm in about 3 to 4 months, and then released egg embryonated egg in feces and complete the life cycle. Pathogenicity. These parasites cause considerable mortality in sheep, cattle, and human. The acute phase occurs during migration of the immature flukes through the liver. It causes parenchymal injury, hemorrhage, inflammatory response, largely mediated by eosinophil. Some larvae penetrate through the liver and diaphragm, ending up in the lung. Patient present initially with fever and hepatomegaly. Later they develop acute epigastric pain, obstetric jaundice, and anemia. Death usually uncommon, but it caused by hemorrhaging in the bile duct. Clinical features. Where cases are symptomatic: diarrhea, upper abdominal pain, malaise, weight losses, fever, and night sweat may begin approximately 2 months following ingestion of metacercaria. The signs of this acute phases of infection are hepatomegaly, splenomegaly, anemia, weaknesses, and marked peripheral eosinophilia. Adult flukes in the bile duct may be associated with cholecystitis and caliculus or acaliculus cholecystitis. Diagnosis. Demonstration of the egg in feces or aspirated bile is the best method of diagnosis. Eosinophilia is constantly present. 3. Serological test by parasitic specific antibody such as immunofluorescence and immunoelectrophoresis may be helpful. Treatment. Oral B-thionyl is the treatment of their choice. Intramuscular imaging has been used successfully. Clonorchis sinensis, commonly known as the Chinese Oriental liver flukes. Clonorchis sinensis was first described in the biliary tract of Chinese in Calcutta. It belongs to the group of Oriental liver flukes, where there are 3 main species which commonly infect humans. These parasites are so similar in their morphology, life cycle, and pathogenicity, and it's parasite of fish-eating mammalian, particularly in Asia and Europe. Man is the definitive host, and water snail and fish are intermediate hosts. Dogs and cats are important reservoir hosts. The adult worm lives in the human biliary tract for about 15 years or more. It has a flat, transparent, spatulate body and brownish in color, pointed anteriorly and round posteriorly, 10 to 25 mm long and 3 to 5 mm broad. They are all hermaphroditic and possess 2 suckers. The eggs are flask shaped and relatively similar in size. They are yellow or brown, bile stained, containing well-developed miracidium. It has operculum at one pole and a small hook-like spine at the other. This slide shows the adult worm of Clonorchis sinensis, in which it contains oral suckers and also contain intestinal and uterus, ovary, testes, and also bladder. The another figure show the eggs of this element. Life cycle. Humans are the principal definitive host, but dogs and other fish-eating canines act as reservoir hosts. They require two intermediate hosts to complete its life cycle. The first one is snail and the second fish. The adult worm lives in the bile ducts of the final host. It contains the ciliated miracidia are discharged in the biliary ducts and passes in the feces. It's usually not hatched in the water, but only hatched when ingested by a species of snails such as Pulimus or Elocinema. The miracidium development through several developmental stages: sporocyst, rhidia, cercaria, in about 3 weeks. The cercaria escape from the snail and swim in water and attach to the second intermediate host fish, usually of the carp family. The cercaria insist under the scale or in the flesh of these fish to become metacercaria in about 3 weeks. This stage considered infective stage for a human. Infection usually occur when such fish are eaten raw or inadequately processed by the human or other definitive host. Frozen and dried fish may act as a source of infection. The metacercaria exist in the duodenum of the definitive host, the adultscaria that come out enter the bile duct where they mature in month and assume the adult form. This figure shows the life cycle of Clonorchis sinensis. The life cycle began when the embryonated egg passes in the feces of the definitive host like a human, and then swim in the fresh water. Then egg are ingested by the first intermediate host, the snail, and undergo several developmental stages: miracidia, sporocyst, rhidia, and cercaria inside the snail, and then fresh swimming cercaria taken by second intermediate host, freshwater fish, and then cercaria undergo insist in the skin or flesh of this freshwater fish, and then metacercaria, when taken and ingested by a human host, can infect the human and exist in duodenum, and then it migrate to the biliary duct and form adult form. The diagnostic stage for Clonorchis sinensis was egg, while the infective stage was the metacercaria. Pathogenicity. Many millions of people become infected every year, but only a minority suffer from any illnesses. The pathology is really related to the number of parasites present. Light infection of up to 50 eggs or more are usually considered asymptomatic infection, while heavy infection of 500 or more eggs may cause serious illnesses. The migration of larvae up the bile duct induce desquamation, hyperplasia, and sometimes adenomatous change. The adult worm may cause obstruction of bile duct and then leading to the cholangitis. Acute infection may be characterized by fever, diarrhea, epigastric pain, and also enlargement and tenderness of liver, and sometimes jaundice. The invasion by this worm in the gallbladder may cause cholecystitis. A few cases go on biliary cirrhosis and portal hypertension. Diagnosis. Diagnosis is made by observing ova in feces following iodine stain. Several serological tests have been described, including gel precipitation and indirect hemagglutination test. Treatment. chemotherapy has not been very successful. Chloroquine and praziquantel have been reported to be useful. Surgical intervention may become necessary in case with obstructive jaundice.